Remember what chromosome stage in mitosis? So these are these prophase chromosomes, metaphase, anaphase, telophase. Now that's a test question, right? Metaphase chromosomes, most condensed, right? We have a male here of the X and Y. Okay. Just a little about the human genome. Based on a publication in, what year is this paper? 2011. So it's a 6-year-old paper, and there's been some updates. But this was published in 2011. The proceedings of the, I'm not sure the journal, but like C.W. Chang et al. 2011. At that point, they discovered 21,000 protein-coding genes in the human genome. So it's just this little bookkeeping of the human genome. Can I erase all this? Are people still copying it, or I can leave it? They're all stunned, you can see. Like, can I change my major? Sure, why not? Let's see. Sewing, that's a good major. So let's do a little summary here. I'll do it this end so I don't have to walk back and forth. So here's some stats of the human genome. Of course, the 23 chromosomes. As of 2011, they discovered 21,000 protein-coding genes. In other words, the nucleotide sequence was expressed all the way via mRNA protein. Okay. They also discovered by then 9,000 RNA genes. Genes that were only, in terms of expression, only went that far. And that's how they function in the cell. Some of these function as ribosomal RNA. Some as transfer RNA. We'll learn about those roles when we get to translation, converting messenger RNA. Notice I got the M here, but I didn't put it here. Okay? There are RNAs that make up part of what's called the spliceosome. Okay? We also have a recent discovery the last few years has been what are called long non-coding RNAs. That's what LNC stands for, long non-coding. Their function has been kind of a mystery for a long time. Now we're recognizing, not we, but scientists that do this analysis, recognize that these long non-coding RNAs sometimes bind back to the DNA to turn genes off. They can help to control gene expression. Okay? Lots of other RNAs out there. All kinds of RNA has a much bigger role than was initially thought. Okay? Of these 21,000 protein-coding genes, about a typical cell, I don't know what that means, a typical cell, will express 30 to 6% of those genes. So, from less than half to slightly over half. So somewhere around 9 to 10 to 11,000 genes are turned on by that cell. The other genes are never used. So genes have to be selected, cells have to be selected which genes they use in a multicellular organism. Even a single-cell organism won't use all the genes all the time. It'll express them when it needs. But there are, of these 21,000, and according to this paper, 2064, I'm sure that number's changed, but at 2011, of the 21,000 protein-coding genes, 2064 are known as housekeeping genes. These are genes that are always expressed because they provide the proteins that allow the cell to perform its basic functions so it can live. These are on in every cell, is what that means. Okay? Every cell. Again, so I've got 23 genes, chromosomes, 21,000 protein-coding genes. That means, each chromosome has hundreds of genes. Some chromosomes have more genes than others. Which chromosome has the most genes? X. It's the most gene-rich chromosome. Which one has the least? Y. Why? Why? I don't know why. Why not? Why? Why? That's why they call it Y. Why? Now, Y used to be big. I guess size doesn't count for the Y chromosome. Actually, it was X and Y were originally the same size. They were originally autosomes that became involved in controlling the sexual attributes of the organism, and Y has eroded over the last several hundred million years. It's quite interesting. Why? Well, I'll show you a short video on Wednesday that talks about the erosion of the Y chromosome. That erosion may be continuing. Not feeling so good. Imagine a world without any males. Would that be good or bad? What do you think? No males in the world? Probably less war, right? And we talked about sex chromosomes, first discovered by Morgan's lab. We've already seen the experiment in 1909, and they found the white-eyed mutant male. Okay, that would confirm sex chromosomes, and that traits could be X-linked. And of course, in humans and in primates and fruit flies, we have the same sex-determining system. It's not universal. It's where if you're XX, you're a female, and all your eggs have one or the other. And of course, one came from a mother, one from a father, so are they identical in terms of the alleles they have? Of course not, unless a woman had a baby with her identical twin, which would be really weird, right? So different alleles on these X chromosomes. Okay, and of course the males have an X and Y, and these are not homologous. They do pair up at prophase 1, but their recombination is restricted right to the tip. Most of it does not recombine because they share very few genes. There are a few genes they share, but mostly they don't. Okay? And which parent determines the gender of the child? The male. Right? There's a photograph of X and Y. Their metaphase state. If we look at X, these are some of the better known chromosomes, better known genes in the X chromosome. And they're, mainly known through their mutational effect when they're mutated. Again, that's how many genes have been discovered. When they don't work right, we describe the mutant and then backtrack to the gene. So there's a protein called dystrophin that's an important protein in muscles, and when it's mutated, it leads to muscular dystrophy. And hydrotic ectodermal dysplasia, that produces brittle hair, and it affects the ectoderm of the skin, leading to brittle hair and other conditions of the skin. Here's a gene that when mutated leads to severe combined immunodeficiency, which if you have this mutation, you have to live in a bubble and you'll die young. We'll talk about this gene in a second, cyst, okay? It's the inactivation control. We'll talk about that in a second. Here are some of the hemophilia genes here. That if you have the mutated version, you don't clot normally. And colorblind genes are here. Okay, if you have a mutation here, you don't produce either the red cones or the green cones. Okay? There are over 1,000 genes on this chromosome. This is just showing some of the more better known ones. Okay? Now, I'm going to just make a few more comments and be back to your exam here. It turns out, if you think about genes, it's important to have the right number of genes for the organism to work correctly. If you have a deletion of a chromosome, a breakage, and you lose that gene. That could be detrimental to the cell. Likewise, if you have an erroneous duplication through replication error, you may have multiple copies of a gene. That happens, that's quite common. And so you can have differences in what they call gene dosage. The more genes, the more protein. That can affect the cell, right? And so think about females with 2 X's and a male with only 1. Since this is the most gene-rich chromosome, there's a difference in gene dosage between females and males. Or there could be, right? You have twice as many genes on the X than you use the Y. And most of the genes on here have nothing to do with gender. None of these have anything to do with gender. Female's the default gender. You only become a male because you got a switch on the Y chromosome that turns female into male. These have nothing to do with being a female, most of these genes. So if you're XX, you got twice as many of those genes. That's the difference in gene dosage between female and male. Make sense? So what happens in mammals and most vertebrates is that as the embryo develops in a female, one of the X's gets turned off in every cell. So ladies, in every one of your cells, you only have one functional X chromosome in those cells. And the cells aren't all the same. Some cells have one X off, some have the other X off. And it's accomplished by this gene here, the X chromosome inactivation control. And what it does, it transcribes a big piece of RNA that combines the chromosome and turns it off, blocks it. Okay? And it's at random. We'll talk about that in the next slide on Wednesday. But what I want to do now is give you a chance to look at your midterm. I've already posted grades, so I'm sure you already know what your grade is.